All right, so I hope you see the presentation in the correct format.
Yes, we see it very well.
Nice. Yeah, so as Katarina introduced, we'll have a webinar series of 5 lectures, and this is the first one. So I will give an overview of what we mean by bioinformatic analysis of single-cell RNA-seq data, which tool we use, and not really how to use them. It's not the purpose of today's topic, but the next ones, yes, because the tools are so different that it's not really possible to cover all of them in one webinar. So that's why we have several webinars. So today's topic will cover the following points. So I will first give an introduction to the single-cell sequencing for people who are not really familiar with this. Then present some single-cell data analysis workflows, talk about the different tools that we use for the data analysis in the single-cell RNA-seq field, and talk a bit about the reproducibility in bioinformatic analysis, which is very important. So the first point is introduction to single-cell sequencing. So when we talk about single-cell data analysis, what we really mean is that initially you have a mixture of cell population that you don't really know what is in there. So you have a mixture of cells. Maybe you have neurons, astrocytes, microglia, if it is a brain tissue, or if it is a lung tissue, pneumocytes, alveolar cells, and so on. But you don't really know what you have there at what percentage. So what we mean by single-cell data analysis is, first of all, to know what is what. What cell is a neuron, or what cell is an astrocyte? This is the first thing that we want to know. What cells we have there? Like, especially when you compare a control versus a disease case, you want to know whether a particular cell type is more present or is it absent. So this is the first thing. What is there? Then we want to know how many of these cells we have. Like, do we have a more, I don't know, T cells because we have a disease, or do we have less of a particular cell type? So this proportion is also very important when we compare different conditions in samples. And then last but not least is which genes play a role in this sample. Like, what makes these cells different from each other? By cells here, I don't mean cells like of different populations, but for example, you might have, let's take the example of the brain again, because I study the brain, so I know this tissue better than any other tissue of the body. So for example, you take the microglia cells that are the white blood cells, the resident macrophages of the brain. The microglia, they can get activated when there is a disease, a neurodegenerative disease. And you want to understand what genes differ, what genes are overexpressed or upregulated or downregulated in this activation process. So for example, here, you see cells that are green, but with different tones, or cells that are yellow with different tones. They are actually cells of the same population, like microglia in green, neuron in yellow, but the tone represents the activation stage. And we want to know which genes play a role in this activation stage. Yeah, so this is basically what we mean when we talk about single-cell data analysis. We want to know all these things. Of course, we can go even beyond. We can do trajectory analysis, like what is the potential fate of a cell, knowing that we have some cells that present a certain gene expression at a specific time. So we can do gene enrichment analysis and so on. But this is basically what we are interested in for a basic analysis. So it is very common to use this fruit salad and fruit juice analogy to compare bulk sequencing, single-cell sequencing. So basically, when we compare single-cell analysis to bulk, in the single cell, you see all the fruits individually, But in the fruit juice, it's a very homogeneous thing, and you don't really see what is in there. So it has one homogeneous color. Is it strawberry? Is it anything else? We don't know. So in the bulk sequencing, we have basically a bunch of cells, like you see here, mixed cells. And of course, what we get is just a blurry measure of whatever we want to measure. For example, if we want to measure the gene expression level of a particular gene, it will just be blurry. So it will be the mean of all the cells together. So we get what we call the average expression profile of the entire sample for that particular gene. For the single cell sequencing case, since you have all the individual cells, you know each individual cell, and I will explain you later how we know that. So you can track all the individual cells, and because of that, you can still measure the gene expression of a particular cell, and then from there understand whether a particular cell population has an upregulation or downregulation of a particular gene. And this helps us identify some biomarkers, for example, of a rare cell population. So the rare cell population, you will never be able to identify them if you use bulk sequencing. It's just lost in this bunch of cells. But in the single cell case, you can identify them even if the number is quite low. So what is the principle of bulk versus single cell in terms of technology? So again, you have a heterogeneous tissue with different cell types here, green, yellow, red. So from this, you get the RNA content, the mRNA, which you convert to cDNA afterwards. So you follow a certain protocol, and then you do the sequencing, and after sequencing, you get what we call the fastq files, which are the output of the sequencing step. So you get the fastq files that contain the sequencing data. But this sequencing data needs to be pre-processed before being able to be analyzed. So at this stage, here we have the fastq files. From here to here, we do the pre-processing. So I don't show the individual steps here. We get here the count matrix. So I will talk about this, what it really means. And it's only after we get the count matrix that we can start to do the downstream analysis afterwards. And of course, for the bulk RNA-seq, we don't really call it count matrix because it's just too small to be called a count matrix. So you have basically a certain set of genes in the columns, for example, and the different samples in the rows. And for each sample, we have the gene expression level. And based on that, you can take, for example, a gene X in the column, whatever it is, and compare sample 1 and sample 2. Well, when I say sample 1, sample 2, I mean condition 1, condition 2, because the idea here is to have the same sample in different conditions to see whether there is a change with different conditions. And when you check the gene expression level for gene X for these two different cases, well, you might see that there is no change. It is possible you might also see that there is a change. Everything is possible. But when you do this with single cell sequencing technologies, so again, you start with the same material, you extract the RNA, but here, the crucial step is that you are barcoding them. So that means that when we extract the RNA of the cells, we barcode them so that we know from which cell they come from. And that's why you see the different colors that are associated with the initial color of the cell. So the red ones come from the red cell, the yellow one from the yellow cell, while here in the bulk technology, you don't have any track to the original cells because everything is mixed. So here you have included a barcode in the RNA or the cDNA, and that's why with this barcode, with the help of this barcode, you can know from which cell it came from. Then you do the sequencing, and again you get a fastq file here that you pre-process to get right now a real count matrix, and from this count matrix you do some other analysis to get the final data. So again, here, if you compare the gene expression, let's say the same gene X in two different conditions, cell type B, cell type C. So you can see that for cell type B, the gene X is actually upregulated in condition 1, downregulated in condition 2, while in cell type C, the gene X does not change. So if the number of cell type C is much, much higher than cell type B, then obviously in the bulk analysis, you will mainly see the cell type C contribution. So you will lose this information. Therefore, it is very important in this case to use single cell technology to understand whether there is a particular cell population that that is modified in different conditions, that is affected in different conditions, in different samples. So this is the idea of single cell sequencing. Maybe to summarize this slide, so in bulk RNA-seq, we extract the whole RNA. So total RNA might be misleading because what we call total RNA is all the RNA content of a cell, including ribosomal RNA. But here, what we are interested in is in the mRNA. So we extract the mRNA, but everything is mixed, so we just get homogeneous cell population. We just get mean expression, gene expression of different samples. While in the single cell RNA-seq, what we get is individual gene expression for the different cells. So in this case, we can detect this heterogeneity within the sample and the cell-to-cell variability. So I mentioned that the main difference between bulk RNA-seq and single-cell RNA-seq was the barcoding step. So how is it really done? Or what is a barcode, first of all? So there are many different techniques to barcode single cells. I am going to talk about maybe the most widely used ones. So you have a bead that's actually very small, bigger than a cell, definitely. It has a metal core. So it will be attracted when there is a magnet in the neighborhood. So it has a metal core and it is coated with some oligonucleotides. And this oligonucleotide has a specific structure. So shown with red, green, blue, and yellow. So the red region is a PCR handle. It is used to amplify whatever binds to here later on. The green region is a cell barcode. The blue is a unique molecular identifier, and the yellow region is the poly-T. It's just a poly-T. So the mRNA, imagine you have a cell lysed, the content, the mRNA content is just released. All these yellow regions that are actually poly-Ts, they will capture the mRNA from the poly-A. So the poly-A tail of the mRNA will bind here. You just hybridise. So all the mRNAs will bind here. One mRNA per one oligonucleotide. The green region, the cell barcode, is the same on all these molecules on the same bead. So one bead has one cell barcode. Another bead will have another cell barcode. However, the blue region, that is the unique molecular identifier, is different from one molecule to the next. So each of these molecules will have a different UMR. So when you have an mRNA molecule that is captured by one of these oligonucleotides, it will receive a unique UMR. So let's imagine that you have a gene that is expressing, I don't know, 10 mRNAs. I mean, this is what is happening. So when we say that a gene is highly expressed, it means that this gene is producing, I don't know, 10, 100 mRNAs in a go, like in a very short time. And all these mRNAs coming from the same gene will bind to these molecules, but to different molecules. So that's why they will receive a different UMI. So in this way, thanks to the UMI, we can know how many mRNA came from a particular gene. Because you should not forget that after this beat capture, we do a reverse transcription and we then amplify all these molecules. That's why you have a PCR handle here. So we do a PCR, and with the PCR, we introduce some artifacts. We introduce many copies of the same cDNA. But of course, those copies will have the same cell barcode and same UMI. So that's how we know that if we have PCR amplicons with the same cell barcode and UMI, then we know for sure that those are PCR duplicates. And we don't count them in the analysis. What is important is to identify the unique UMIs or unique mRNAs that came from the gene to have a better estimation of the gene expression level. So this is basically how it starts, the beat capture and the introduction of the cell barcode. So there are several technologies that use this kind of beats. The droplet-based method, where you have the cells that are introduced from a certain inlet, the beats are introduced from another inlet. So this is an aqueous solution here. And then a cell and a bead will get close to each other. And from another inlet, you have oil. So because of this oil aqueous solution interface, you have a small droplet forming. Oops. And the bead and the cell will be captured in the same droplet. And then everything else will happen in this droplet. So the cell lysis and so on will happen in this droplet. And the mRNA will be captured by the beads later on. And we'll get the barcode. So after the mRNAs get the barcode, of course, these mRNAs will be reverse transcribed to cDNA. So we should talk about cDNAs. Once the cDNAs have received the barcode, we can mix all the cells together. There is no problem because we have barcoded the individual cDNAs. So this is one way. Another way that is used in our company, Signal Biotechnologies, is to it's a microchip-based method. So we basically put, you see the microchip here. We basically introduce the cell suspension from one of these inlets. So it will flow inside, and the cell will just deposit or precipitate to the bottom of the well randomly. Of course, the cell suspension should be dilute enough in order not to have doublets in the same well. And then we introduce the beads in a high density so that for sure we have one bead per well. You will not have more than one bead per well because of the size of the bead and the size of the well. So once this is done, then the cells are lysed and the content, the mRNA content, will be captured by the bead. So here it is captured in the droplet, while here it is captured in the well. This is the main difference. Okay, so once we have our cDNA barcoded, then we just follow the protocol, we get the libraries, and we do the sequencing to get our sequencing data. So the next step, or the next topic of this presentation, is to talk about the single-cell data analysis workflows. So the bioinformatic workflows can be divided into two main categories. The first one is the pre-processing, and the second one is the downstream analysis. The pre-processing starts with the sequencing data, which is the fastq file that I mentioned. Then it goes through several steps that I will not detail today. It is a topic of the next webinar. So it goes through a series of pre-processing steps. And at the end, we get a count matrix. A count matrix is a matrix in which we have for each cell and for each gene the UMI count. So how many UMIs do we have for a specific gene in a specific cell? And as I mentioned, the UMI, the unique molecular identifier, is an indicator of how many mRNAs a gene has produced, if we capture all the mRNAs, of course, which we don't. So we cannot say that if we have an UMI of 100 in this count table, that gene produced exactly 100 mRNA. This is not correct. We just capture less, of course. Okay, so we get the count matrix, and once we get the count matrix, so for each combination of cell and gene, we get the counts for this particular gene. We use this as an input to our downstream analysis, and this is where we try to get meaningful results, like biological insights. Okay, so that's why this first step is the pre-processing step. We just prepare the field, prepare the data, so that we are ready to analyze in the downstream analysis. Two main workflows, and of course the tools that are used for these different parts are also different. So that's why I needed to talk about these two separately. So for the pre-processing part, as I said, the input is a fastq file, and the output is a count matrix. So if you just focus on the fastq file here, it looks like this basically. So it has a header starting with X, with some information about the flow cell and so on. Then the second line, it is the sequence. And then you have a quality score here, shown with some characters, F or comma, etc. So basically, this is one line, starting with an X. And the sequence with A, C, A, etc., etc., wrapped to the next line is the second line. Then you have the plus, and then you have the fourth line that is again wrapped. So the number of characters that you have in the fourth line and the number of characters that you have in the second line are equal. So this F represents the quality score of this A here. Now, what does it mean to have an F here? Well, I don't know by head what number this represents, but basically you have a table that shows you like what is the probability to have here not an A. So we are confident that this is an A at 99.99%. Okay, so this score, called the Fred score, represents this probability. But we need to look in a table to understand really the probability. But the character itself, I don't know by heart. It's just a shortcut to this probability use. So of course the probability is low, like the error probability is high, then the Fred score is low. Okay, so this is only for one, this is called one read here, the sequencing that you see, it's only the sequence of one molecule, and you have millions of molecules that are sequenced. So this is only for one. From this data, fastq file, we get this count matrix. So we get the sequence, we do some pre-processing, we clean it, and so on. And after alignment to a reference genome, we see where it is aligned, at which genes it is aligning. Because we know the barcodes, we can know to which cell it belongs to. So you see cell 1, it is aligning to gene 1, then we put here a unit of 1. If we have elsewhere in this fastq file another read that comes from cell 1 and aligns to gene 1, then we increment this value by another unit, and so on. So that means that there are 5 reads that come from cell 1 aligning to gene 1. Basically, this is how you would read it. And similarly for other other cells and genes. But as you see, you have many zeros. So you have cells that do not have any alignment on some genes, plenty of zeros. So in this case, having this kind of count matrix is really not optimal because you just store a lot of data that do not give any information. You basically store a lot of zeros. So another way of storing this is what is called the sparse format. So we just consider only the values that are non-zero. So only the red ones. So then you arrange this in a slightly different way. You have 3 columns now, for cells, for genes, and for the counts. So for cells, you have cell 1, which is this first row. Then you go to gene 1, which is here, you have 5 counts. Then the next one would be for cell 1, gene 4. So here I should have put 4 and then count 2. So here there is a mistake. It should be 4 here. And same for the rest. Okay, so in this way, you basically save some memory. So your initial data that was maybe more than 500 megabytes will become 50 megabytes. I think this is true for 1000 cells approximately. If you have higher number of cells, of course, your data will be also higher. But this ratio is more or less a factor of 10, something like that. So yeah, it saves you a lot of memory. So this was for the pre-processing part. And now you have the count matrix, whether it is a full matrix or sparse count matrix, it doesn't really matter. As I said, it's just to save memory, but working with both is okay. So once you have your count matrix, you are ready to do further downstream analysis. So what are we interested in? Well, we would like to know, for example, what cell types we have there in our sample. So identification of the cell types first. Whether there is a difference between samples in the amount of these different cell types. So for example, here you see the different cell types that have been identified. Like here, for example, dotlet cells are located here, teft cell, enteroendocrine cells, and so on. So here you have 2 data together, 2 sample data together. And if you plot them for the individual samples, you see that from sample 1, most of the cells are actually in this region. They're coming from this region. You see here, it shows the cell density for sample 1. And for sample 2, you have this population that arises, and this one as well at the bottom. So then, whatever the condition in sample 2 is, you understand that goblet cells seems to play a major role there, or the enterocytes as well. So this is an intestine sample, as far as I see from the list of cells that we see. And then when you also look in the gene expression file, you can also see that the gene expression is not the same in different conditions. So this is the kind of analysis, the kind of result that we want to get. And we can even go beyond and do some enrichment analysis or even trajectory analysis, which again will be for the next webinars. So this was an overview of the single-cell technology and the main workflows in bioinformatics. So now, which tools are used in single-cell data analysis? So again, starting with the pre-processing part. So as I said, you have the individual steps here in the pre-processing, and each of these individual steps, they have their own tools. And those are usually command line tools. So you need to use them by just typing commands. So if you are familiar with Linux, basically those tools are written for Linux environment. If you are familiar with Linux, with this black window, like terminal environment, you will need to type these commands in this window. Of course, you can make them automated by writing all these commands one after the other in a file and then run the whole file. You can do that. So this is already a first step in writing a pipeline or a workflow. However, When you write a pipeline, you have some requirements. First of all, it should be able to run several samples in parallel. So if you have 20 samples, you should not run each sample sequentially, but it should run everything at the same time. If you have the resources, if you have a HPC, high performance computing system, you need to use that so that everything is done faster. So you need to also write the pipeline in a way that parallel computing is possible. Another important requirement is that it should be able to adapt to available resources. For example, here, the different steps will require different resources. The alignment, for example, there are different alignment tools, but one of them is star, and star aligner requires 32 gigabytes of RAM, which is a lot. And so it needs 32 only for itself, but the other steps do not need 32 gigabytes. So it would not be smart to accommodate 32 for the whole pipeline and run this sequentially, because then 32 gigabytes will be reserved for each of the steps that are run sequentially anyways, just because this part needs 32. So you need to be able to adapt for the individual steps. So each step might require different RAM resource, and you need to be able to give this to each individual step. And another requirement is that you should be able to run the pipeline. And if for a certain reason the pipeline stops because of an error, and you need to rerun it after correcting the error, then it should not regenerate the files that are already there. Otherwise, you will just waste your time. For example, let's imagine that you provided the location for a genome file that is used here, but this location is wrong. You just made a typo. Then you run everything correctly from here to here, perfectly, no error. But then here it shows you an error message. Then what you do, you correct the path and you rerun it. It should not rerun everything. It should just start from where it stopped. This is another requirement of a good pipeline. And for this, there is no reason to reinvent the wheel. They are already workflow management systems. So I am citing two of them here. One is SnakeMake and one is Nextcloud. And there is another one called Cromwell, which I have not shown here. But yeah, those are the two widely used workflow management systems. And I'm going to go through each of them now. So the Nextflow community, so SnakeMake and Nextflow, they are the workflow management tools or languages. And anyone who uses this tool, the Nextflow, for example, to write a pipeline that is ready to use, he will upload his pipeline on a website called NFCore. So when you go to the NFCore website and click on pipeline, you will see this kind of list. And maybe you can find your happiness somewhere, like here. Maybe you want to use this scRNA-seq pipeline. Let's see if it is for you. I mean, it already gives a certain description, a single cell RNA-seq pipeline for 10x genomics data. So if you are not using 10x genomics data, then this pipeline will not work for you. But if you do, then you can click on there and see the detailed description. Yeah, and then here it tells you how to use the pipeline. So the only thing that you need to do is to download this, install whatever you need to install, and follow the instruction to run it. So you don't have to worry about the individual step that I showed previously. Like here, you don't have to worry on how to do the multiplexing, trimming, etc., etc. Everything is already done for you in those pipelines, but you just have to know which input you have to provide. Okay, and there are many different pipelines for many different things that you can do. You just need to go through them. and see whether one of them is matching your need. Yeah, so this is about Nextflow and the Nextflow community. They use the NF core to upload their pipelines. Now, coming to SnakeMake, this is slightly different. So there is the SnakeMake community share their workflows mostly on GitHub. So there is no SnakeMake workflow website. However, there is what we call a SnakeMake wrapper repository. So here you have a SnakeMake workflow. Well, we call it a wrapper, actually it's more correct, for the individual steps of the pre-processing. So again, if I go back, each of these steps, you will find a SnakeMake wrapper for these. So the only thing that we need to do is to take these wrappers and put them next to each other in a file to make your pipeline, basically. So that's it. And what is the advantage? Well, the advantage is that you can customize the pipeline as you wish. You just need to use the wrapper, so you don't have to go into the details again. You don't have to go into the details on how to use the trimming or the alignment tools. You just have to use the SnakeMake wrapper and put them in your file. And you can use in this way different tools. Like, again, if I take the example of the alignment, there are many different alignment tools. And if you don't want to use star, but I don't know, any other, HiSet2, for example, then you can use the SnakeMake wrapper for HiSet2, for example. Okay, while with the Nextflow, since the pipelines that are shared on the NF core, they are pre-made pipelines, you cannot really modify them unless they provide the option of using different alignment tools in their pipeline as an input. So this is the main difference between the two. Okay, so that was it for the pre-processing part. Now we are at the downstream analysis. So in the downstream analysis, the tools that people widely use are the Seurat package in R, developed by Rahul Satija lab. And what you can do with this, well, you can have a detailed information. If you go to this website, you can do transcriptomics and epigenomics analysis. It's not only transcriptomics, not only RNA-seq, but also ATAC-seq or DNA methylation. I'm not sure, I did not check that, but you can do epigenomics analysis. And yeah, so basically you can do the conventional analysis with cell annotation, differential gene expression analysis, enrichment analysis, trajectory analysis, and so on. Then you also have the option of doing multiomics. So in case you have RNA information plus any other information at single cell levels, for example, protein information at individual cells, or epigenomic information in individual cells, you can combine both RNA and the other extra layer modality. So here, each of them is called modality. Protein would be one modality, RNA information would be another modality. And you can combine several modalities together to do more accurate analysis, because it is possible that the RNA is degraded very fast, but the protein that has been produced from that RNA is more stable. So you will capture the protein information, but you will not capture the RNA information. So by using both at the same time, you get a more accurate identification of the cell type. So this is also important. This you can do, multiomics. Then what else can you do? You can also do spatial transcriptomics. So in this case, of course, the technology is slightly different. You don't have a cell suspension that you put from an inlet and then it will be captured by a bead, etc. This is totally different technology. You have a tissue section, basically, on a slide. And I'm not going to enter in details, but when you put this tissue section on the slide is covered by some oligonucleotides with a poly-T tail that will capture the mRNA as soon as you lyse the tissue. So you lyse the tissue on the slide, and at the location where you see these dots, here. It has a certain size, and the size is the same everywhere. So basically, at this dot, you have many oligonucleotides with a poly-T, but it has the same barcode within a dot. So one dot will have the same barcode. And this barcode is associated with its spatial location. So, and this way, when the cells are lysed and the mRNA is released and captured by this oligonucleotide, local oligonucleotides, based on their barcode, we can know where on this slide they are coming from. So we can map them back and then map the gene expression at this particular location. So this is also what can be done with the SORAT package. Then, for people who prefer to use Python, there is a scMPy package. Again, you can check this website for full documentation. You can basically do the same thing as what SORAT offers you, but maybe with some more possibilities. So first of all, so the scMPy package is developed by the group of Fabian Theiss and the LMU. And here you see many packages developed around scMPy. So they are all compatible with scMPy. And this is called the scVerse. So you can again check this website for full documentation of these individual packages. But basically, each of these individual packages is an extra tool on top of scMPy to do what you would like to do. For example, SquidPy is what you would need, the extra libraries that you would need to do spatial transcriptomics on top of scMPy. Muon is the extra libraries that you would need on top of scMPy to do multi-omics analysis. SIRPy is to do T cells and B cells analysis, for example. The SciTools is an extension of scMPy to do more statistical analysis. And here, those are just data formats compatible with scMPy to facilitate the easy access of the data, manipulation of the data, etc. Like the SORAT object in R, here the equivalent format would be the annotation data or AND-data object. And the new data object is basically a combination of AND data object coming from different modalities, like one coming from RNA, one coming from protein, etc. So this is called the new data. It's a combination of AND data. And the new data is used with Muon to do multiomics. AND data is used with scMPy directly. Okay, so this is the complete set of packages that you have in your hand to do the Gaussian analysis. So I think many people are interested in a comparison between SORAT, scMPy, or maybe R Python. Of course, there are people who are fan of R, people who are fan of Python. I mean, everyone has his own reasons, and they are right. Because when you go through this table, you will understand quickly why. So when you look in statistical tools and plotting tools that are available in R, you have much more options than when you compare it with Python. So in Python, if you need to do a plot similar to R, maybe you need to write several lines of code to do the same thing in R, while in R, it's already a ready-to-use function. It's already there. In the multi-omics and spatial transcriptomics, there is no real difference, I would say. So both of them are available in both packages. However, when it comes to machine learning, you have more limitations in R. It's much more advanced in Python because of the structure of the programming language itself. And the speed is also faster in ScanPy and slower in Seurat. This also has been shown. And on this next slide, I will even show you how one can integrate, one can use the GPU to even accelerate things further. So I don't know whether you have heard about this, but there is Rapids, which is an ensemble of Python libraries that use GPU. And this is compatible with the ScanPy package. So what is a GPU, first of all? So this is more computer architecture, I would say. So basically, most of the people are using the CPU. So you have here, you see 4 cores. The CPU is 4 cores, and you can because you have 4 cores, that means you can run 4 tasks in parallel, not more. 4 tasks in parallel. If you have another CPU next to it with again 4 cores, then you can run 8 cores in parallel. A GPU, however, is actually something used in graphic cards. And graphic card has many pixels, millions of pixels, that you see here, the individual pixels. And the idea of using a GPU is basically to launch tasks on each of these individual pixels in parallel. And it speeds up things very, very much. Of course, there is this difference between CPU and GPU. First of all, well, as I just mentioned, in the CPU you have low number of cores, while in GPU you have a much higher number of pixels. However, the processing speed in these cores is higher in CPU and lower in GPU. So if the parallelization that you're looking for is not so high, then using GPU will not accelerate things very much. It really accelerates things when you go very, very to the edge, like millions of cells to be analyzed, for example. Then launching the calculation in parallel on each of these pixels will accelerate things. And you can see here a comparison for the different steps of the analysis. The second column shows the time in second for the CPU-based architecture, and the next 3 columns show the times in the GPU architecture with 3 different models. Let's not care about the models for the moment, but just see how what the acceleration factor is. For example, calculation of the t-SNE takes 220 seconds in CPU architecture, but only 2.8, 1.4, or 2.2, depending on the model of the GPU, seconds here. So you see the acceleration factor here. In some cases, it's not really affected, like the pre-processing step. You see it's even slower in some of the GPU models. The UMAP calculation is again accelerated by very much in a GPU-based system. The clustering is also accelerated with a GPU-based architecture. The differential gene expression analysis, again, also. So overall, when you compare the timings, so end to end, you see a high factor of acceleration when you use a GPU architecture. And as I said, Rapids, it's a Python library, an ensemble of Python libraries that are compatible with the ScanPy package. So you don't have this with the Seurat. So if you are looking for, first of all, machine learning, and on top of that, you are looking for using a GPU-based system, because in machine learning, people are using GPU, then you need to switch to Python if this is your goal. But if your goal is to do conventional data analysis with very sophisticated plotting and statistical analysis tools, then using R is better. Yeah, so this would be the conclusion here. So which interface do you need to use for downstream analysis? So of course, since you use either Seurat or ScanPy, you would use either R or Python, which means you would use RStudio or PyCharm or Spider, etc., as an interface. However, it is a very good practice to use a Jupyter notebook because it can be used with both R and Python. First of all, you can write your script in there and run it directly in there and have the figure also embedded in there. You can write some explanatory text. So of course you can write it in your scripts by using the comment indicator, the comment symbol. But here it really looks like a report. So you can even share it with your colleagues. He will see the explanation, he will see the code. You can also hide the code if you want and show only as if it was a report. And here the philosophy of Jupyter is that it's a cell-based interface. So what you see here is a cell. And you put your code in the cell and you run that cell, and it will give you an output. And you can run Python script in one cell, R script in another cell, in the same Jupyter notebook. This is possible. So it has a bunch of advantages, I would say, to use Jupyter rather than sticking to RStudio or any Python user interface. Okay, now coming to the reproducibility in bioinformatic analysis, it's a very important concept because you will realize, or you have already realized, that there are a lot of lack of reproducibility, and for several reasons. So if you want to reproduce the same data that you have read in a paper published a few years ago, even if you look in the methods, you go through the methods, you use exactly the same packages, etc., you don't get the same results. Why? So some of the points are obvious, some of the points are less obvious. First of all, using different tools, Seurat or ScanPy. This one is obvious. If you do your analysis in Seurat and in ScanPy, you will not get the same results. You will not get the same visualization of the results, let's say. Okay. So of course, at the end, the biological insight will be similar, but you will not get, for example, the top 10 differentially expressed genes will not be the same. The order will be different. And the reason is, if I take this as an example, it's very simple because of the way Seurat and ScanPy calculates the log-fold change to identify the marker genes. In Seurat, it basically takes the exponentiation of the log transform gene expression, and then it calculates the mean. So at the end, what you get is the mean gene expression. But in ScanPy, it first calculates the mean of the log transform gene expression, and then it takes the exponentiation, which at the end is mathematically equivalent to this. So the product of the gene expression to the power of 1 over n. So mathematically, the one that Seurat is using is an arithmetic mean, but what ScanPy is using is a geometric mean. And geometric mean is less sensitive to outliers. So if you have some outliers, then your marker genes will also be different. But which one is correct? We don't know. At the end, it's a mean, whether it's arithmetic or geometric, it's a mean. So I don't know whether it is correct to say one is more correct than the other. It's just a different method. But you need to bear this in mind if you want to know why the list of marker genes that you obtain with Seurat or ScanPy are different. This is one of the main reasons. So another reason of lack of reproducibility is because you use different versions of the same tool. So ScanPy 1.7 is different from 1.8, etc. So it will also produce different results. Presence of a random variable in the tool, and this is true for several algorithms like UMAP, Leiden, Luma for the clustering. So when you run these tools again on the same data, just at a different time, you will get different results. Of course, sometimes you will not see the difference, but in some cases you might see, especially when you do the clustering. The clustering will just be different because these algorithms, they use a stochastic method. it is the nature of this algorithm. Stochastic method means that it uses a random variable. And the random variable that the computer uses is based on the computer time. So if you run the same script at a later time, the random variable that the algorithm will take will be different, and your result will be different. Usually, or most of the time, you don't see this change, but you might. So if you want to keep the reproducibility, you need to set the random variable seed that is provided as a parameter in those tools. Usually, this is skipped in the analysis that are done in several publications, and it's not even provided. Like nobody provides which random seed they use for their analysis. So that's why you will never be able to reproduce the same plots. Using a different database, of course, this is also obvious, or using different filtering parameters, but usually those are provided. So some of them are controllable, some of them, it depends on how you work. So really, if you pay attention to the details, you also need to take this random variable thing into account. At least things that you can control is the version of the different packages that you use. And therefore, it is highly recommended to work with Conda environment, for example. So you install everything in a Conda environment so that anything that you would install outside this environment will not affect your installation within this environment. So you can have another Python version or R version outside this Conda environment, and then it will not affect your environment itself. However, if you want to provide this Conda environment to another person, you need to provide an environment file with a list of scripts that are loaded in this Conda environment. But sometimes it is possible that another person will not get the same installation because you are running on Mac OS and he is running on Linux or Windows. I don't know. So in this case, it is safer to use Docker containers or Singularity containers. So to use containers specifically. A container, it's something that is standalone. So it has its own operating system. It is completely closed to the outside world unless you allow it. While the Conda environment is not closed to the outside world. So you have access from your Conda environment to any file of your computer. But in a Docker container, once you are in there, you don't have access to any file outside unless you specify at the beginning when you launch it. Okay, so you have these different possibilities to make sure that you don't lose reproducibility. Working with containers is very important. And you can also find different containers, ready-to-use containers with already pre-installed bioinformatic workflows on Docker Hub. So if you go to the web page Docker Hub, you will see there a list of ready-to-use Dockers with pre-installed pipelines in there. So you don't need to install anything by yourself. You just need to download it and launch it. And everything is already installed. Same for Singularity. So Docker is more popular than Singularity. The main difference between these two is that Docker, you are super user within the Docker. So you can do whatever you want when you are inside. But usually, universities that give access to the servers or HPC systems, they have Singularity installed and not Docker. Because with Singularity, if a user has certain restricted privileges, he will have the same restricted privileges when he is connected to the Singularity container, inside the Singularity container. So he cannot install anything as he wants once he is in there. But with a Docker, if he has restricted privileges on the server and launch the Docker, there it has the super user privileges. So that was it overall, but if I may summarize very quickly, I basically mentioned the difference between bulk RNA and scRNA. So this was done with the cell barcoding. Use of workflow management system for pre-processing data is very important, such as SnakeNet and Nextflow. Use of Jupyter Notebook for downstream analysis. You can use R and Python in there. And Docker or Singularity containers for reproducibility is also very important. So that was it on my side. Thank you very much for listening.
Thank you very much, Adam, for this very informative webinar. I think there were a lot of questions that people had. I think there is a person that would like to talk to you. So let me allow you to do that.
Okay.
Hello.
Yes.
Can you hear me?
Yes, we can hear you.
Yeah.
Okay, thank you very much for this great talk. I'm not really familiar with the single cell transcriptomics, so therefore I wanted to attend the webinar. Maybe my questions are naive, but I wanted to ask. So as I understood, this barcoding is very important point when we are doing this. And can experimenter or the user design the barcodes by himself or herself, or are they available on the market? The second question is, how many barcodes should be used to identify the cell type in a safe way? Because, for example, I am also a neuroscientist working in brain, and for the microglia, we have several markers, could be also only by microglia or but expressed also in the macrophages. So we should be very careful. So therefore, to be in the safe side, how many barcodes you use? And the last question is for the spatial transcriptomics. You said that you are lysing the slice, the tissue on the slide. And if we are talking about the brain cell, how thick should be? So this is the questions that I would like. Thanks.
Okay, 3 questions in one go. All right, so the first one was, can we or can anyone produce their own barcodes? Yes, it is possible. If you have the tools for that, sure. I know some labs that they were producing their own barcoded beads, but usually if you buy the kit from a particular company, they provide the beads, the barcoded beads. So this is one thing, but of course you are allowed to design your own barcodes as well. It's up to you. Second thing is, how many different barcodes you need? Well, in the single-run technologies, I think we have many different barcode combinations. 384 to the power of 3 is already a lot, right? I cannot calculate this, but it's several millions. So that means that you can capture several millions of cells. But usually, you never put several millions of cells on this microchip. It has an upper limit, like 10,000, something like that. Or if you go for high density, it's even a bit higher, 30,000, but it's never millions. So that means that if you want to capture the rare cell types, then you would need to run several chips to reach these millions of cells. So you need to have an estimation of the percentage of those rare cell types. So if you think that the percentage of this rare cell type is 1%, that means that over 100 cells, you will have 1 cell of this population. So from 10,000, you will have 100 and so on. So 1% is okay. But if it is even more rare, like 0.01, then you need to calculate this and see how many chips you need to run. So this was the second question. The third question, you said, when you place the tissue section on the slide, what should be the thickness? Well, this again is provided by the protocol of in the kits of the different companies. And I believe there is a lower limit, first of all, because you will cut the cell into half. So you will lose some mRNA content. So you don't want that. But you also don't want to have it too thick, because if it is too thick, the mRNA after cell lysis will just diffuse sideways. And because of that, you will just have a mixture of the mRNA of neighboring cells. So you will not be really accurate. And if I need to give a value, the thickness is around 5 microns to 20 microns. It's between these two values, 5 to 20.
There is maybe one more quick question that we can ask because we are running over time. So is there a difference between the Python and R packages that can be used for the pre-processing analysis, or how do you see the situation?
For the pre-processing analysis, you don't really use R or Python for the pre-processing because it's a common line tools. So the R or Python is not really involved in there.
And later on during the analysis, is there any difference between those two?
Yes. So as I also mentioned in the reproducibility part, the differentially expressed gene that you will detect will be slightly different because they use different calculation methods.
All right. Thank you very much. Thank you very much for all the attendees. And thank you, Adam, for giving this very informative talk. We will be happy to welcome you again next week, next Monday. I wish you a successful week, and I'm looking forward to meet you again next time. Thank you very much.
Thank you. Bye-bye.
Bye.